Welcome, everybody. This is a special seminar. This is our, our usual Monday research rounds, but today is actually one of the BHI Brain-Heart Interconnectome research seminar series. And the purpose of this is really to explore this interconnection, interdisciplinarity, and the kind of transformational types of research that we're doing that is at the brain-heart interface. So, of course, very closely aligned with the BHI activities at the university. And our speaker today is Dr. Jason Fish, who is absolutely going to show us a very cool, very innovative story that is exactly that at the brain and heart interface. And for those in the room, as we know, we're going to do questions at the end, and I'll moderate. I'll run around with the microphone, and then we always have a good presence online. So online, please feel free to ask questions using the chat function. Dr. Fish is a senior scientist at the Toronto General Hospital Research Institute and the University Health Network, and a full professor in the LMP program at U of T. He did his PhD at the University of Toronto under the supervision of Dr. Phil Marsden. And that's where Jason's work uncovered a role for epigenetics in gene regulation of the vascular system. And, we maybe all take epigenetics a little bit for granted at this time now, but at that time, it was really innovative to know that there were sort of supragenetic regulations that were required for blood vessel development. So this was work that Jason did during his PhD. Then he went on to do just a minor training at the Gladstone Institute with Deepak Srivastava at the Gladstone at the UCSF. And that is where I think Jason and I probably first overlapped and interacted, because Jason really discovered some very cool roles for microRNAs in cardiovascular development. So since then, Jason and I have had the pleasure of working together on a number of projects exploring microRNA functions, but Jason's lab overall studies the regulation of gene regulation in the endothelium. He's discovered recently how somatic mutations in the endothelium lead to some sporadic brain AV malformations, a leading cause of stroke in young people. And his laboratory is uncovering mechanisms and information to design new therapies accordingly. And so this is, again, an example of how we can understand at the molecular level what's happening either in development or when things go wrong, and also to try to leverage that to understand, how we can better treat these diseases. And one of the interesting things Jason has been doing a little bit more recently is studying the role of extracellular vesicles. And so between epigenetics, microRNAs, and EVs, Jason could not have chosen 3 more technically challenging things to study. And so at each turn, he must think, how can I make this a little bit harder? But thankfully, because he's not only a brilliant scientist, but he's got, a great team always behind him, he's always making strides and pushing it even more forward. So thanks for accepting our invitation, Jason, and please welcome to the podium. Thank you.
Right. Well, thank you very much for that very kind introduction. I'm happy to be here and present some of our work. We've been working sort of in separate domains in the lab, in blood vessels in the brain and in the heart, and these things are sort of slowly converging together. What I want to share with you today are a couple of stories based on extracellular vesicles in the heart and the brain. And talk about some future directions we're going in terms of where we see this EV-based communication going. So you probably are aware of extracellular vesicles. I'll just have one sort of background slide on these. These are small membrane-bound vesicles that are secreted by most cells, and they're very much involved in communication. So there's a lot of different types of vesicles that can be made, including pointer here. It's all right.
Then it should be able, we can hide.
I was trying to point to things, but I can't really. So there's different different types of vesicles. There are exosomes that are made from multivesicular bodies, as well as microparticles or microvesicles that actually bud off of the membrane. And these different vesicles, there's a whole variety of these. The reason that they've become very interesting is that they have different contents, and there seems to be an active loading of different material like proteins, nucleic acids, including microRNAs, and lipids into these vesicles. And they can be secreted from one cell type and either act locally and get taken up by a cell type in the environment, or these can actually travel in the blood and be taken up in distal locations. So we've been quite interested in this as a communication strategy, and a rich source of biomarkers and trying to gain insight into biology. So when we think about the endothelium, it really is at a location in the body where there's a lot of different signals coming from different areas, from the blood, from the tissue. And the endothelium, we think of as a real communication nexus. There's a lot going on in the way the endothelium communicates with tissue, but then how different components in the blood communicate with endothelial cells and other cells in the microenvironment. So when you think about where the endothelium sits, it's lining blood vessels. A lot of these vesicles that get made by the endothelium end up in the blood, and they're quite stable in the blood. So they circulating around in the blood, and if we actually take those out and look for biomarkers, this can tell us something about the health of the cardiovascular system, particularly the vasculature. Vesicles that are produced by the endothelium or that are received by the endothelium can modulate communication, and that is through a number of different processes. But there can be transfer of microRNAs between one cell and another, as well as protein-mediated transfer or signaling. There's also a great potential, I think, in terms of these as being used as therapeutics or as therapeutic targets. I won't talk about that as much today. I'm really going to focus more on the first two bullet points. In the lab, we have a number of projects that are looking at EV-mediated communication, and I could have chosen a number of those to talk about. I wanted to focus on a couple that are on this one theme of how diabetes actually affects this process. But we have other projects where we're looking in atherosclerosis at how EVs are potentially mediating plaque vulnerability. We are also interested in how the endothelium and cardiomyocytes are communicating by EVs in the setting of cardiotoxicity. That's kind of an emerging area in the lab. But what I wanted to focus on, just to have some thematic organization to this talk, is I wanted to focus on diabetes as a real systemic disease that affects a lot of different organs. But we want to focus in on how extracellular vesicles might be involved in this pathology. So we want to look at how diabetes is affecting the brain vasculature, and how EVs might be involved in being a biomarker of cardiovascular disease, particularly diabetic cardiomyopathy. So I'll start with focusing in on the heart. And in particular, diabetes is known to really elevate the risk of heart failure. A large percentage of diabetic patients will actually develop some form of cardiomyopathy that is varying severity. And this is associated with a lot of structural changes in the heart. So you will get left ventricular hypertrophy or dilation, depending on the stage of disease. It's often associated with fibrosis, so that the heart becomes stiffer. And really the reason that we initially got into this was because of the association with microvascular rarefaction. So when you look in the left ventricle of the heart, there's actually fewer microvessels in the wall of the heart. And that's actually what intrigued us initially to look at this particular disease. One of the things that diabetes in particular is associated with is diastolic dysfunction. So that's of course an issue with the relaxation of the heart. This is the consequence of having a stiffer heart. And so there are associations, pretty strong associations with heart failure with preserved ejection fraction. The challenge with HFpEF is that a lot of the early stages are quite asymptomatic. So it's very difficult to identify patients that have diastolic dysfunction because it's often asymptomatic. And a lot of diabetic patients have diastolic dysfunction and just don't know about it. So there's really a need for biomarkers of these early defects that happen. And we've been interested in trying to understand whether the cardiac microvasculature might provide some of these biomarkers. So if you look at a cartoon of the pathology of this disease, there is, as I mentioned, defects in the microvasculature in the wall of the heart. And this is associated with an increase in inflammation and endothelial dysfunction and capillary rarefaction. And one of the driving factors may be reactive oxygen species. As you will know, diabetes is often associated with more reactive oxygen species. And in the endothelium, this can lead to less nitric oxide production. And nitric oxide in the heart is known to communicate with myocytes. And in the absence of this signal, there's an increase in stiffness and hypertrophy. So the endothelium is directly communicating with myocytes through this process. There's also, during the course of the disease, a lot of fibroblast activation and fibrosis that occurs to really contribute to the stiffness of the heart. So we were interested in trying to understand what the mechanisms might be for microvascular dysfunction in the heart, and whether we might be able to find circulating biomarkers of that microvascular dysfunction. And these, I think, are quite interrelated questions, as you'll see. So there are a number of models that we could have looked at, but we chose to use DB mice, which are leptin receptor mutant mice. These are very obese mice, and they very quickly develop obesity and hyperglycemia and insulin resistance. You can see even by 6 weeks of age, these mice are clearly diabetic. This was work that was initially started by Sean Beach and Sebastian and Jacques in the lab. So one of the things that we found in characterizing this particular mouse model is that they develop a progressive diastolic dysfunction. So at 8 weeks of age, they have pretty normal diastolic function, but by 14 weeks of age, they develop diastolic dysfunction, as you can see here by looking at strain. In parallel to this, we looked at the vasculature in the heart, and what we found was that there is a defective microvascular density quite early on. So even earlier than we see diastolic dysfunction, by 8 weeks of age, we see less density of the microvasculature. There's also an increase in mean lacunarity, and what that means is just the spaces between vessels. Those tend to increase. So if you look in the left ventricle, it's poorly vascularized compared to non-diabetic controls, and it actually happens quite early. And this has led many to think that maybe the microvascular dysfunction is actually one of the primary events that happens even before you get cardiomyocyte dysfunction. If you look at the endothelium, it's also, there's a lot of markers of damage to the endothelium. At the top here, we're looking at 4-H&E. This is a lipid peroxidation marker. And you can see that by 8 weeks of age, this is quite high, specifically in the endothelium. So there's a lot of oxidative damage that is happening in the endothelium of the microvasculature, and this persists out to later stages of disease. There's also evidence of DNA damage and senescence. So if you look at phospho-H2AX, this is a marker of double-strand DNA breaks. It's also thought to be a marker of senescent cells. And you can see that, again, at 8 weeks of age, when you have microvascular rarefaction, you also have more double-strand breaks in the endothelium. One of the really major breakthroughs in terms of therapy for HFpEF is SGLT2 inhibitors. And there's lots of interest in these drugs. And this really stems from a couple of New England papers a number of years ago now that showed that these drugs can actually decrease the amount of heart failure and cardiovascular death in patients with HFpEF. And up until this point, this was the only drug class that could actually make any impact on HFpEF. So this has been quite an exciting area. We have used these drugs in our model of diabetes. And this is work by Corey and Rafna in the lab. And we use empagliflozin as an SGLT2 inhibitor and actually add this to the diet itself. And you can see that quite early on, you get hyperglycemia in these diabetic mice, but EMPA actually maintains normal glucose levels in the blood. Despite not having any effect on the weight of the animals, it is actually improving glucose handling. What we found is that these drugs actually work quite well in modifying the defects in the microvasculature. So you can see that if you take a mouse and treat it with EMPA, that you can rescue a lot of the microvascular rarefaction defects. Now, we wanted to use this system to really try and get at what was happening to the endothelium at the molecular level that might change function. And so we actually, in this mouse model, we isolated endothelial cells from the left ventricle and did RNA sequencing and attack sequencing to try and understand pathways that might be affected. When we did RNA sequencing, we could see that surprisingly, most genes that were regulated were downregulated. There were very few that were actually upregulated. There's just a massive downregulation of more than 600 genes. And when we actually started looking at what those genes might be, a number of these were angiogenic factors, including transcription factors like SOX17, AP1 transcription factors, SRF, and other transcription factors that are known to drive angiogenesis. And then, of course, things like vascular endothelial growth factor and semaphorins. All of these genes are involved in angiogenic signaling. We found that when we looked at the pathways that were regulated, these were involved in things like axon guidance, kinase signaling, and VEGF signaling. So there's clearly an effect on angiogenesis, and these SGLT2 inhibitors seem to improve that. Looking at the attack sequencing, and this is a bit of a complicated slide here, but this is just showing the changes in transcription factor footprinting in the endothelium. And so what you can see with this is that in mice that were treated with EMPA, there's actually an increase in transcription factor families like AP1, ETS, MEF, and SOX factors. And these are all known to be involved in angiogenesis. So these are increased by EMPA. And then on the other hand, there is a decrease in transcription factor footprinting for hypoxia-inducible factor and other pathways that are involved in endothelial to mesenchymal transition. So there's a lot going on in the endothelium at the molecular level. But I started off by saying at the beginning that we wanted to find biomarkers that would indicate what's happening in the microvasculature. So this is helpful because we know what's happening in the microvasculature. But if you thought about a patient that you had that you might suspect would be at the early stages of this disease, it's not like we're going to go in and actually do a biopsy of the heart. We really need something else to use as a biomarker. And this is really where the extracellular vesicles come into play, because these are potentially quite helpful biomarkers that are circulating in the blood. And we're looking for signatures that might inform what's happening in the cardiac microvasculature. So here, what we've done in this experiment is use that same mouse model at either early stages, 8 weeks, or later stages, 14 weeks. And we've isolated extracellular vesicles from the blood. from the plasma, and then looked at the size and quantity of those vesicles. And what you can appreciate here is that in diabetic mice, there's more vesicles made, and they tend to be larger. That's just quantified here at the bottom. And this is a fairly early change. By 8 weeks, there's already changes in the circulating pool of vesicles. We wanted to go more in depth in terms of what was in those vesicles. And we've worked for a number of years in microRNAs. So this is where we looked initially for potential biomarkers. We used a qPCR array to profile 100 microRNAs, and then looked for ones that were changed in the setting of diabetes. There were a number that were changed significantly. I want to focus in on the miR30 family. We were able to validate this using a different technology, using microfluidics. And then we also used a totally separate animal model. In this case, these are a lean diabetic model in rats called GK rat. And we found a number of these microRNAs were similar between the rat model, which is a lean model of diabetes that also develops diastolic dysfunction, and the mouse model, which is an obese diabetic model. We confirmed that the miR30 family was increased in the setting of diabetes in both the mouse and the rat model. And interestingly, these microRNAs are induced in these EVs in circulation quite early, and it coincides with the changes that we see in the cardiac microvasculature. Now, interestingly, when we looked back at the RNA-seq data that we got from isolated endothelial cells from the heart, a number of targets of the miR30 microRNA family were altered. So in diabetic mice where there's increased levels of miR30 in circulation, there's also a decrease in miR30 targets in the endothelium. So that was our initial clue that maybe this is reflecting something that's happening in the cardiac microvasculature. Now, of course, when you're looking at a sample from the blood and looking at EVs, there's a lot of different places that those EVs could be coming from. A lot of different cells, a lot of different organs are secreting extracellular vesicles. So we wanted to get some insight about where this signature might be coming from. And when we looked at miR30, we found that it was induced in the left ventricle of the heart, and really wasn't changed a whole lot in a panel of other organs that we looked at. When we pulled out the endothelial cells from the heart, and I'm showing you here PCAM, which is a marker of endothelial cells to show that we can actually enrich endothelial cells from the heart. We found that the miR30 family, miR30D and E, were specifically induced in the endothelium of the heart, not in non-endothelial cells. So this seems to be an endothelial response to diabetes that miR30 is upregulated. And as one does with microRNAs, the next thing you would want to do is try and understand what the microRNA potentially does. And so we looked at prediction of target pathways. As anybody who has done this in microRNA field, you end up getting hundreds of potential targets. But what was quite interesting when we looked at the pathways that those microRNAs were predicted to target, there were a couple that really stood out: fatty acid biosynthesis and fatty acid metabolism. And there's a number of potential targets here that I'm showing to the right. What was interesting about this was that one of the earliest changes that happens in diabetic cardiomyopathy is a shift towards fatty acid metabolism. It's a really early event. That's thought of mostly in terms of cardiomyocytes, because cardiomyocytes shift almost exclusively to fatty acid metabolism in the setting of diabetic cardiomyopathy. But this was interesting because this is a pathway that popped up with this particular microRNA that's induced by diabetes. So we wanted to look at pathways that this microRNA might be regulating in the endothelium. And so we used approaches where we could overexpress the microRNA or knock down the microRNA family. And you can see here that these inhibitors can inhibit miR30D and miR30E. And then we looked at this panel of predicted targets in fatty acid metabolism pathways. And in endothelial cells that are challenged with palmitate, so that's a fatty acid, a lot of these genes are induced. So it really induces a number of these genes that are involved in metabolism of fatty acids, which makes sense. If we overexpressed miR30 before we treated cells with palmitate, this suppressed the induction of a number of these genes. We confirmed a couple of these genes, FADS1 and ELOB-L5 at the protein level. And then the opposite in cells where we inhibit the miR30 family, we found a more robust induction of these fatty acid metabolism genes. And again, at the Western blot level, we also saw that these two genes were targeted. So this was quite intriguing that this microRNA induced in the endothelium affects fatty acid metabolism. There's been a lot of work done over the years and really some interesting discoveries over the last several years about how the endothelium regulates cardiac metabolism. It's known that myocytes use mostly fatty acids as a fuel source. And of course, that they're constantly needing fatty acids as a fuel source to maintain contraction. But most of that fatty acid actually comes from the blood. And it has to cross through the endothelium in order to be utilized by the cardiomyocytes. One of the main ways that this gets into the myocytes is through the liberation of fatty acids from triglyceride-rich lipoprotein particles. And so lipoprotein lipase, which sits on the endothelium, liberates this. It then goes from the endothelium to the myocytes. And for a number of years, it was just thought that this was just almost like a passive process, that the endothelium would take up these fatty acids and would transfer them to the myocytes. But work from Zolt Irani's lab and Bill Sessa's lab has shown that actually it's quite a dynamic process. There's a lot that happens within the endothelium. It's not just a passive passing along of the fatty acids, but the endothelium actually processes these fatty acids. And there's quite a dynamic conversion of these fatty acids to triglycerides, formation of lipid droplets, which is interesting to some people in the audience, and then further processing towards fatty acids that are then transferred to myocytes. So there's a lot happening within the endothelium. So we wondered, with this microRNA regulating this fatty acid metabolism, what this might do to this process. In endothelial cells that are challenged with fatty acids like palmitate and oleate, they form lipid droplets. And if you overexpress miR30, you could probably appreciate from here that they actually form more lipid droplets, and that is quantified here to the right. So there's something going on in terms of fatty acid biology. I'll say that we don't quite fully understand all of the lipid droplet biology here, but it really pointed to something that this microRNA is doing to metabolism. Now, if we come back to this schematic, the endothelium not only just transfers fatty acids to the cardiomyocytes, but can also use those fatty acids. So the endothelium is mostly glycolytic. It uses glucose as a fuel source, but can use fatty acids to create ATP through beta oxidation. So we wondered whether miR30 might affect that process. And this is a seahorse assay, which looks at basal and maximal respiration using the fatty acid palmitate as a fuel source. And you can see that under basal conditions, and the endothelial cells overexpressing miR30 use fatty acids more to create energy, which is measured here by oxygen consumption rate. And this is even greater when you look at maximal respiration. And that's just quantified here to the right. And if we do the opposite, where we inhibit the miR30 family, they use fatty acids less as a fuel source, and that's quantified again here to the right. Now, the endothelium using fatty acids as a fuel source is good for creating energy, but it's not a process that's without a downside, and that is the creation of reactive oxygen species. So if we look at cell ROX, which is a measure of oxidative stress, when you challenge endothelial cells with palmitate, there's more oxidative stress, as you might imagine. But when you have miR30 and they're actually using fatty acids more as a fuel source, there's more oxidative stress. There's also more 4-H&E, which is a lipid peroxidation byproduct. So there's more oxidative stress when you have more miR30. The other component that I mentioned at the beginning is nitric oxide production, which the endothelium is very important in creating nitric oxide, and it signals to underlying myocytes. Interestingly, palmitate itself downregulates eNOS expression quite dramatically. miR30 overexpression also does this, and if you combine those, you get an even further reduction in eNOS. And you can see that at the protein level as well. This effect of fatty acids on eNOS, Zolta Rani recently had a really nice paper showing that this is mediated through effects on lipid droplet formation. If you look at eNOS in the heart, you could also see that at early stages, eNOS is maintained, but at later stages, you get a loss of eNOS mRNA. So we think that this is likely important in some of the vascular effects that we see. So as a final experiment for this particular project, we wanted to see if we could actually rescue some of these effects by inhibiting miR30. We used global inhibitors of miR30, these LNA inhibitors, and we injected over the course of several weeks at 5 weeks, 6 weeks, and 7 weeks, and then looked at 8 weeks of age. And what we found is that the density of the vasculature was somewhat rescued. Particularly the spaces between the vessels were narrowed, which suggests that there's better microvasculature. But the more striking thing that we saw was that markers of oxidative stress, like 4-H&E or DNA damage, the phospho-H2AX, were reduced when we decreased miR30. So what we have learned from this particular project is that in the setting of diabetes, a lot happens molecularly in the endothelium. I didn't show it here just for the sake of time, but we do see evidence of senescence occurring in the endothelium, and we think this is really important in inducing miR30. miR30 gets packaged into extracellular vesicles that we can actually detect in circulation as a marker for what's going on, we think, in the cardiac microvasculature. We haven't looked at what miR30 does in those EVs in the setting of the heart, but there could be something interesting there. And this miR30 family seems to target fatty acid metabolism. It decreases fatty acid biosynthesis, but at the same time seems to increase fatty acid beta oxidation. We don't fully understand all of the processes that are going on there, but the end result seems to be more oxidative stress and DNA damage in the endothelium, which we think drives endothelial dysfunction, microvascular rarefaction, and eventually diastolic dysfunction. At the same time, we've also found that the endothelium itself has a downregulation in angiogenic genes. So you get less microvasculature and less ability to form new vasculature through angiogenesis. And SGLT2 inhibitors seem to improve that part of the disease. So I'll shift then from a focus on the heart to look at what's happening in the brain. And diabetes is known to have a really profound effect on cognitive health and affects the blood-brain barrier. And that can then lead to cognitive decline and dementia. Well, what we were interested in knowing is whether, you know, this has been well documented for a long time, that diabetes affects cognition, but it's really not been well understood why this is. A lot of the assumption is that this is because of hyperglycemia, maybe advanced glycation end products and other damage. There really hasn't been a lot of focus on the vesicles. And when you look at I've shown you some of this already, that in mouse models of diabetes, there's a huge increase in the number of vesicles. This also occurs in diabetic patients. So in the blue and yellow, these are human patients with and without diabetes. You can see there's a lot more vesicles, and these vesicles tend to be larger. We wondered what these vesicles might do to the endothelium and integrity of the endothelium, because with all these vesicles floating around in the blood, the endothelium is constantly exposed to these vesicles. And what we were surprised to find, this is using Excelligence, which measures the integrity of the endothelium, and in a dynamic fashion, so you can actually measure this over time. What we found is with diabetic vesicles that were isolated from diabetic mice, if we put those on an endothelial monolayer, a confluent monolayer, it very quickly disrupts the integrity of that monolayer. We were surprised with how rapid this was. We're very interested in microRNAs, and we initially thought, you know, maybe microRNAs are being transferred from these vesicles into recipient cells, but this is really way too quick for that to be the case. This is a very rapid change in barrier. We used a separate, more quantitative measure of vascular barrier integrity by looking at the permeability of Fit-C dextran. And in both mouse harvested vesicles or from patient blood, this disrupted the monolayer either early at 1 hour or later at 24 hours. And if you use proteinase K to remove the surface proteins on these vesicles, it mostly got rid of the effect. So it seems to be something about the protein content of these vesicles. We also looked on the right at human patient samples, and we saw the same thing. It disrupts barrier early on, and that barrier disruption is maintained long term. As I mentioned before, SGLT2 inhibitors are quite promising therapies. And we found in our mouse models that treatment reduces particle size and EV concentration. And also, if you take those vesicles, they are less able to disrupt vascular integrity. So potentially, there's a lot of interest in SGLT2 inhibitors in terms of improved cognition in diabetics. It may be partly through this process. We collaborated with Philippe Monier's lab at Toronto Western. He uses intravital microscopy using a cranial window in the mouse. And you can actually inject fluorescent tracers and look using two-photon imaging at leak in a dynamic fashion. So these mice at the top are non-diabetic, the bottom are diabetic mice. They're injected with two different sizes of tracers, a 10 kilodalton tracer and a 70 kilodalton tracer. The 70 kilodalton tracer really doesn't leak out unless there's major damage to the endothelium, like a hemorrhage where you actually get loss of blood, whereas the 10 kilodalton tracer will leak out when you have subtle changes in permeability. What you can see from this is that the diabetic mice have a lot more of the 10 kilodalton tracer leaking out. It's quite dramatic leakage. What we did was actually then take circulating vesicles from diabetic mice or non-diabetic mice, and then injected those into a non-diabetic mouse and looked at the permeability of these cerebral microvessels. And what we found is just taking the vesicles themselves and injecting them into a non-diabetic mouse could disrupt barrier. So it suggests that these vesicles are actually quite potent at disrupting barriers. We looked here at the brain microvasculature. We haven't yet looked at other vascular beds, but certainly this is quite an important vascular bed for this disease. We then went on to try and understand what these vesicles might be doing. We used phosphoproteomics to get at this and looked after just an hour of exposure to vesicles and found a number of pathways of interest, disruption of cell-cell junctions, lamellipodia, actin cytoskeleton, cell-cell adherence junctions, et cetera. And in terms of signaling pathways, the RAS-MAPK pathway really was overrepresented. Now, when you look at the endothelium, there's different ways that things can cross through the endothelium. There's paracellular leak, where substances will move between the endothelium, and then transcellular leak, which we'll look at in the next slide. So what you can appreciate here is that V cadherin is an adherence junction that really is between all of the endothelium. It forms a very nice barrier. If you TNF stimulate these cells, they lose barrier. And on the right, the diabetic vesicles could decrease the intensity of V cadherin. And shown with the arrowheads, you can actually see spots where V cadherin is missing. Claudin 5 is a tight junction protein that's really important in barrier. And again, with the diabetic vesicles, it could downregulate Claudin 5. So there is clearly some effects on paracellular leak. With Warren Lee's lab in Toronto, he's developed a technique to look at transcellular leak. So this is movement of substances through the endothelium. And here we use a fluorescently labeled albumin, and it will actually move through the endothelium. Measure this using turf microscopy at the basal membrane. What we found is that the diabetic vesicles also increase transcytotic events. So there's paracellular leak and there's transcellular leak that's happening when the cells are exposed to vesicles. And finally, we wanted to try and see if we might be able to target some of those pathways. The ROA ROCK pathway is really important in modulating leak, and it does that by affecting actin cytoskeleton. So when you have ROCK being activated, it phosphorylates myosin light chain, and you actually get contraction of the actin cytoskeleton that induces leak. What we found is that the MAP kinase pathway and the RO-ROCK pathway were activated so that we could see increased phospho-ERK and phospho-MLC2. And in the bottom plot here, you can see that it's quite rapid. So really within 30 minutes, you get MAP kinase activation from vesicle treatment. And the final experiment that we did was to look at whether if we pre-treated with a MEK inhibitor or a ROCK inhibitor, we actually block the permeability induced by these vesicles. And indeed we could. So the diabetic vesicles induce more permeability, but inhibiting either the MAP kinase or the ROCK pathway could block this effect. So what we found in this particular study is that the vesicles that are circulating around in the blood have a major impact on the integrity of the vascular barrier. And in the setting of diabetes, these vesicles are changed, their contents are changed, and their function changes so that they induce paracellular and transcellular leak and disrupt the blood-brain barrier. We think that this might be important in the setting of diabetes-induced cognitive issue, particularly with vascular cognitive impairment, that it's really impacting the microvasculature in the brain. Now, what I want to do just in the last few minutes is, so those were two major stories that I wanted to tell you about how EVs are involved in both the heart and the brain. I wanted to leave you with a couple of concepts that we're thinking about in the lab now. If you think about the endothelium, it really is very much a polarized layer of cells. It sits at one side facing the circulating blood and the other the tissue. And so if you think about how the endothelium communicates with these different compartments, so, cells in the blood versus cells in the tissue, this really requires polarized communication. So if you think about how an endothelium might communicate with a circulating monocyte, that would be apically secreted vesicles that might interact with those cells, whereas communication with smooth muscle cells and macrophages would involve basolateral secretion of vesicles. So this was a concept that we had, and this is work done together with Catherine Howe in Toronto. This was kind of the concept that we had, and a lot of the work that has been done in this field is often very reductionist. It's using, co-cultures, isolated vesicles in a very reductionist fashion. But we wanted to test whether the secretion of vesicles is actually polarized. Does it matter? And do endothelial cells actually secrete different vesicles in both directions? To do this, Sneha Raju, a graduate student in the lab, used a very large number of transwell plates to be able to do these experiments. Anybody who's worked with vesicles knows that you need a lot of cells to do this. So she had an incubator full of these transwell plates, and this allows you to isolate vesicles from the apical compartment versus the basolateral compartment. And we made sure that there was an intact monolayer present. What she found was that the vesicles that were made in both of those compartments were different. So there were more vesicles that were secreted apically. If you activate the endothelium, they made even more apical vesicles. And using EM, we could actually see these multivesicular bodies that were positioned either close to the apical side or the basolateral side. This is in cultured cells, but we could also see this in mice, that we saw multivesicular bodies that were really positioned for apical secretion versus basolateral secretion. Really interestingly, when you look at these vesicles, and we did both proteomics and microRNA sequencing, they were quite different. So there's really different things that are being packaged into these vesicles and the apical side versus the basolateral side. And that really is kind of a new paradigm, because it really suggests that the cells are actively packaging different things for secretion in one direction versus the other. And finally, we found that those vesicles, either apical vesicles or basolateral vesicles, could modulate cell types that they interact with, like monocytes and smooth muscle cells. So I really just wanted to leave you with that concept that the endothelium might secrete vesicles in a very polarized manner. And I think this is important because if you think about the heart or the brain, in the heart, the endothelium is very much interacting with cardiomyocytes. Cardiomyocytes are directly interacting with endothelial cells. That would require basolateral communication with myocytes. If you think about the brain, the cells that make up the blood-brain barrier, there's a whole variety of cells that the endothelium is surrounded by that these EVs might communicate with. And so I think that this concept is going to be really important in understanding how these vesicles work in their microenvironment. And the last data slide that I want to show is just some tools that we are building, Catherine Howe's lab and my lab, to try and visualize this process. It's, as I mentioned, a lot of these studies have been done using very reductionist approaches, just because it's very difficult to get enough EVs to really work with. We've developed a transgenic mouse where we use an endothelial Cree driver, an inducible Cree driver, to activate a CD63 GFP reporter. So CD63 is a key component of vesicles, and this is tagged with GFP. So whenever you have Cree, you get expression of this transgene. And you might be able to appreciate here, this is in an atherosclerotic model with LDLR knockout mice over 12 weeks of high-fat diet. You could see GFP expressed in the endothelium. But if you look deep within the plaque, you can also see GFP, which are labeling vesicles within macrophages within the plaque. And so we think in the setting of atherosclerosis, this is quite a nice tool to look at endothelial to spoon muscle cell and macrophage communication. But these types of tools also could be quite useful in, the heart or the brain to look at how these cells are interacting. So just to summarize then, I've shown you today that circulating EV microRNAs can be quite useful in not just as being a biomarker, but actually getting at some of the biology. In this case, it helped us really look at this miR30 pathway in the setting in the microvasculature and HFpEF. The circulating vesicles interact with a lot of different tissues. And what we've seen in diabetes is that these vesicles can disrupt the integrity of the microvasculature. And then finally, this concept that the endothelium is really a nexus for EV communication. So they can secrete vesicles in a polarized fashion, and they also are recipients of vesicles from different locations. And although I didn't really look at this specifically today, I think you can see from looking at all these pathways that EV-mediated pathways could be targeted therapeutically. So just I'll stop there and thank the people that have been involved in this work. Some of these projects were actually initiated from the Canadian Vascular Network years ago. So that's kind of what seeded a lot of the initial work in this area. And we've had other grants from CIHR and Ted Rogers as well. Thank you.
Thank you. Thank you so much, Jason. That was excellent. Just going to check online here. I'm going to ask the first question if you don't mind. So first of all, that was a beautiful talk. And having worked in a little bit of these fields, I can appreciate the technical challenges associated with all of the work that you've done. So bravo to you and the team. I've never really, is there a consensus whether it's the secretion of EVs to communicate to another cell that's the most important, or is it the secretion of an EV to get rid of whatever's in there and it just happens to communicate with another cell? Do you have a, speculative hypothesis, and just thinking in the context of your work in diabetes, is that an injured endothelium getting rid of some of the injury, or is it a signal for help?
Yeah, it's a good question. Like initially with the field, like when these were first discovered, it was sort of thought these were just garbage dumps, right? Like right. They just, cell wanted to get rid of something and it just got rid of it, and that was a way of getting rid of waste. A lot of this seems like when you look at the function of the vesicle, seems fairly purposeful. Like it's not. I guess it could be a side effect if you just wanted to get rid of something and it has an effect on a cell. But it seems from this, when you look at it and start to drill into the mechanism, that there does seem to be some purpose to that, that it is actually a communication. But there could be examples, and I've seen some of this work in the setting of cancer, where actually there are microRNAs that actually repress proliferation. And when a cancer cell secretes these, they then proliferate.
Right.
So I think there is also examples where cellular biology, like a cell wants to get rid of something just so that it
right.
gets something else.
Yeah.
So
because I was thinking that with
it's probably not a
either or. Yeah.
thing.
Yeah. So I was thinking with your the fatty acid oxidation and biosynthesis, is there a scenario where the miR30 is leaving and de-repressing something because the cell needs something versus, anyway, just.
Yeah, it's hard to say. Like, I guess if you overexpress something in a cell, probably more of it will also get secreted just because there's some generic pathways that secrete. So I don't know with the miR30, whether it's just because miR30 is getting induced, that you just happen to get some that gets secreted that you can measure as a biomarker.
Right.
Versus it actually, we've never really looked at the function of miR30 in the vesicles.
Right.
Is it actually doing anything, or we've just approached that purely from really a biomarker perspective, that we can see this kind of leaks into circulation.
And I think it's always challenging because you're right, when you overexpress things do just come out, like even those, but under endogenous conditions, it can be very selective. So there's sort of a lack of tools, but also bravo for making that tool, the CD63, endothelial cell is great. The next step, if I can request, is to make it so that you can tell if it left the cell where it went, right? So rather than just be, because then you can do sort of autocrine, paracrine, so if you could do that next, that'd be great. Thank you.
Yes.
Problems. Jason is busy enough as it is. Yeah, so this is wonderful. And especially I love the concept of the polarization of the endothelium. And so then the EV could actually regulate locally, in terms of neighboring cells. But it could also then release some of these, to actually do more remote type of, coordination or communication, so make it almost like the endothelial cells can be both kind of a signal recipient as well as a signal transmitter, in type of setting, so this is really very, very exciting. So a couple questions. So one is, it's fascinating that the EV from a diabetic mouse model have such a, sort of dramatic and very a time, sort of intensive effect. Question is, did you look at what's in those EVs, besides microRNA, that actually can have such a dramatic effect? And of course, the $1 million question that people don't really, have an answer to is, what is SGLT2 inhibitors doing? Is it metabolic or is it something else altogether?
Yeah, so like in terms of what's in the vesicles, we did proteomics. Although that paper was published quite recently, a lot of that work it's been a long time in publishing that work. So when we initially did that proteomics, we were using an older pipeline for proteomics. So we got a lot of things that were changed, but nothing that was really sort of an obvious pathway to chase after. So we haven't done that. We've now, working with Elena Akawa's group in Boston, she has a really good pipeline for proteomics, specifically of EVs, because there's a lot of sort of optimization that you need to do to look at an EV proteome. So I kind of suspect if we went back and revisited that with the new approach, that we might get more insight. We didn't really get a whole lot of pathways that we wanted to look at. There were definitely things that were different, but they weren't, obvious things to go after. The phosphoproteomics was looking at the recipient cells and what pathways were activated, which was actually quite useful to us, because that was looking, regardless what's in the EV, like what do they actually do? I think with anything related to EVs, it's going to be a lot of things. Like, it's probably not one protein in the EV. It's probably a whole network of things that have an end result on the cell. In terms of the SGLT2, I don't know that I have much insight into that. I mean, the only thing that I say is that there's really not much, there's not much SGLT2 expressed in the heart, like in the myocytes or the endothelium. So it's probably not a direct effect in the heart itself. It's probably something indirect, whether that is just better regulation or whether it's something more systemic metabolism. I don't know that I have any insight into that yet.
But we do agree, because we work on some biomarkers that are very similar, like IGF-BP7 or NGF-2, etc. And so very, very parallel stories. Yeah, and so absolutely.
Beautiful work, Jason, as always. So in looking at these EVs, are you thinking about the lipid composition of the membranes and how this may be changed in a diabetic setting? Because I'm wondering, with the rapid effect that you have on the, compromised barrier integrity in the span of 30 minutes, do you think that's something within the EVs that's, you know, or an immediate effect of just altering membrane composition? And then in thinking about this, you know, is the membrane composition of the apical secreted EVs distinct of that of the basolateral ones? And does that play into cell specificity of the recipient cells?
Yeah. Yeah, we wanted to look at like lipidomics, things on the vesicles, but we've never really explored that. But I'm sure that there must be major changes. Lipid composition, especially in diabetic, there must be differences there. We've just never looked. Same with the sort of apical versus basolateral. It was incredibly challenging to get enough material for the proteomics and microRNA sequencing, and lipids were on our list, but just never could get enough material. But I think there must be something there, and it probably is. The big question, I think, is like, you know, how you get a vesicle secreted. How is there actually any specificity of where it ends up? Like, is there a zip code that sends it to a particular cell, or is it just whatever cell it bumps into? Probably lipids and proteins are going to be important that lipid that directed uptake, which I think there's some evidence of that, particularly in the cancer field. That, you know, particular cancers, actually the vesicles get taken up by the tissue that the cancer often metastasizes to. So there must be some specificity.
Thanks.
Very nice work, Jason. Your penultimate schematic got me thinking a little bit. I mean, you've done a lot of work with endothelial cells, but obviously the EVs are interacting with other cells in the blood, right? Monocytes, neutrophils. I'm wondering if you have any evidence, or, you know, have you ever treated PBMCs with EVs? Do they have any heightened response? So do they have like enhanced chemokine receptor signaling or MMPs, so that you almost have like a two-hit scenario where the EVs are directly affecting the endothelial cells? You have that permeability, but now not only that, but you have the circulating cells that are now more primed to actually act on that.
Yeah, so a number of years ago, we looked at how healthy endothelial cells produce vesicles that dampen monocyte responses. And we only focus on a healthy endothelium. So like the endothelium will secrete vesicles. In that case, we were looking at NIR10A, which is transferred to monocytes and suppresses NF-κB. So there we have that was 10 years ago that we've done that work, but we never really looked at the dysfunctional endothelium, whether it then primes cells for responsiveness. You know, I think it would, and I think there's some examples of that in the literature where there does seem to be like the activated endothelium will activate other like monocytes or macrophages through EVs. So there must be something there. We haven't taken it to that next step. Good.
Beautiful work.
Okay. Okay, great talk. Thank you. Do we know the half-life of the mirrors inside the EVs?
Yeah, that's an important consideration. Like the turnover. So if you think about an EV circulation, they actually turn over, I think, quite rapidly. So if you just take EVs and inject them into the blood, a lot of them get taken up in the liver and actually removed from circulation like within minutes. So there is, so I guess there's two parts of that. Like one is actually the turnover of these themselves, and then the microRNAs in these. The microRNAs in EVs tend to be quite stable, like they're protected from nucleases that are found in the blood. So it's thought that they stabilize the microRNAs when they're encapsulated in the vesicles. I think the bigger turnover is probably the vesicles themselves, that they actually just don't hang around in circulation very long before they get taken up by the liver. So I think what's interesting about that, though, is that what it means is when you're taking a blood sample, you really are looking at a snapshot of what's happening right then and there in the individual. And if you look like an hour later, you'd probably be looking at a different set of vesicles. So it's very dynamic. But I think that creates issues too. Like if you think about biobanks, often we don't think about like If you get a patient, they come in whenever they come in, morning or evening or whenever, and they get blood sample taken. They might have just eaten, they might not have, it's morning, there might be circadian rhythms to all of this. So it creates some challenges in terms of biomarkers as well. These vesicles could be quite different depending on when it was.
So when the mirrors are packaged into the EVs in the source cells, is it possible that some of the nuclease may also be packaged together? I mean, another question is, when these EVs go through the kidneys, the glomerulus, how much of them may be lost in the urine? I mean, how much will remain in the blood stream?
There's definitely some that gets into urine so that you can use, the urine as a source of biomarkers for what's happening systemically. So yeah, there's a lot of points at which the vesicles get taken out of circulation. So they are very dynamically turned over, which again, like if you think about EVs as therapeutics, that's a challenge, right? Because if you inject something, you want it to have an effect, say on the heart, but most of it goes to the kidney or the urine, then you're kind of losing a lot of your therapeutic. And that's where there's a lot of interest in trying to direct where vesicles go. Like, can you, decrease the amount that gets taken out through the liver so that they might go more preferentially to where you want them to go? I think there's a lot of work.
Great work, Dr. Jason. My question is more of a general question. So in the experiments you used, where you used endothelial cells, cultured endothelial cells, how can you confirm that these cells actually respond the right way, since these cells need the hemodynamic component? These cells need the flow. So how can you confirm actually that they really do respond the right way?
Yeah, so I would say the way to look at these experiments, because we always have to use reductionist approaches to look at cell biology. So it's kind of saying that this is how EVs could affect biology, but it's hard to then take the next step to say, like, this is absolutely how they work. Yeah, I think introducing, like, we've thought about how to introduce like hemodynamics into this, because that's a key component. Those experiments are quite challenging, but they are possible. Just we haven't sort of introduced that next. But I think it's a good point. And even just, if you're looking even at, you're interested in the cardiac microvascular endothelium, we can take those endothelial cells out of a heart and grow them in culture, but they change even when you put them in culture, right? They're not exactly what they're like in the heart. So I think it's always important to think about those questions.
Yeah, thank you.
Okay, fantastic. Thank you so much, everybody. Thank you to Jason for accepting our invitation. He's now off to give another presentation because he's very famous. So thank you so much, Jason.